Alright, so I'm going to be doing a little quick video here on the polymerase chain reaction, or PCR, of GMO versus non-GMO DNA found in strawberries. A little bit of an introduction here. Like I had mentioned, it's strawberry DNA, and we had 4 different sample types. We had a positive and negative GMO DNA control, as well as 2 samples of our strawberry DNA that we had extracted previously that I have another video on. For this sample, we had also had a positive and negative indicator in our strawberry DNA to determine whether or not it was GMO positive or negative following PCR. For our tube setup, you can see here I have the negative control, which is non-GMO, positive control here, GMO DNA. Our sample, along with the positive master mix, this is GMO positive indicator, as well as the GMM, which is the negative indicator. For the placement in the PCR machine, you can see here there's another lab groups, but ours are highlighted here. You can see the plus, minus, and then the 2 GMM, PMM. You can barely read those there, but then there were 2 samples of each, so that way we had extras in case there was some type of issue when we ran into it following PCR. For our device settings here, you can see we have 3 stages. You have the heating up process here. We up 94 degrees Celsius. You work down to 59 degrees Celsius, back up to 72 degrees Celsius, and then end at 4 for the whole process. You can see that the volume was for 50 milliliters as well. Here's another setup. You can see here we're starting our running process. So this is 3 hours and 15 minutes beginning at stage 1, which I had said before is 94 degrees Celsius. So throughout the entire process, I know I had mentioned the stages before, but the difference in temperature is kind of explained here, which I'll run through. What you're doing here is by heating up the DNA to a certain degree, you're causing a level of denaturation. So that's going to actually separate the 2 strands, breaking the supercoil and kind of exposing the base pairs here, which you can see. Once the temperature is dropped back down, the annealing process begins and allows the primers to base pair in complementary fashion to each of the strands that were separated earlier during denaturation. And then the increase in temperature again leads to an extension period where polymerase is able to extend the primer, and it's going to form this DNA strand that you're seeing broken off here. This is going to continue over and over and over as these cycles progress, and you can see it's an exponential curve in terms of how many copies of DNA are being made. And this is able to exponentially quantify the DNA so that we are able to do other tests on it and ensure its presence. So in conclusion, I do not have any data yet regarding our PCR, whether or not the GMO versus non-GMO DNA was successful. I would assume that with the increase in agricultural technology and increase in demand for fruits, that these strawberries are going to be GMO positive as GMOs begin to flood the market. I do not think that that is necessarily a negative thing. However, I think it is important that it is expressed while buying the products. As long as there are no altercations in the effects on the strawberry in this case that we're ingesting, I don't think that it's a huge deal, but I think it is important to be able to carry out these techniques and replicate these techniques when we're ensuring the foods that we're consuming. So thank you for watching my video here, and have a good one.